All right, well, thank you for having me here this week. This is really exciting. I want to thank the Simons Institute for putting this on. Oops.
Your mic is off.
My mic is off. Sorry about that. Better? I think we should take a second and see how remarkable it is that we're having a 4-day workshop on large-scale computational theories of the brain. This doesn't happen very often. A little bit of an indicator of that is our instructions as speakers was to encourage us to be somewhat speculative. And if you're a theorist, that's what you're supposed to do. But in neuroscience, it's actually often looked down upon. So I think it's great that we're here, and I really appreciate the Simons Institute for doing that. I'm going to talk about a large-scale theory of the brain. I'm going to talk about the neocortex today, and it's a great follow-on to Surya's talk, which was great. I'm going to talk about a theory about how the neocortex itself may be organized around locations. And specifically, I'm going to propose specific neural networks in the neocortex where grid cell-like mechanisms, not grid cells per se, which are in the entorhinal cortex, but grid cell-like mechanisms representing locations exist in every cortical column, and that the cortex can be understood as a location processor. That's a lot to cover here, and I'm going to get into it. I'm going to start with is this working? No, it didn't work. Oh, there we go. Just what you may not know Numenta is. Numenta is a privately held research lab, a company. We're in Redwood City, California, and we have about 12 employees, and we have a scientific mission. And our scientific mission is to reverse engineer the neocortex. That's arguably a very ambitious goal, I understand that, but it's also a realizable goal, and we shouldn't forget that. We're interested in biologically accurate theories. We're not interested in biologically inspired theories. We're not interested in sort of advancing machine learning. We want to understand how the neural pill that exists in our heads actually works, and we test our theories empirically with partnerships and via simulation. We do have a secondary goal, but it's definitely secondary. We don't spend as much time on it. We believe that cortical theory will be the basis for future machine intelligence. And we have an active open source community who implements our theories and applying them into machine learning tasks. Okay, so let's just get into it. I'm not sure why this is not very reliable. I'm going to start with a quote from Francis Crick of DNA fame. And the latter part of his life, he became interested in neuroscience, and he was totally consumed with the ideas, how is it the neurons in our head could perceive? How it is we have the scene in front of us, and how could neurons be doing this? He wrote an essay in 1979 that was very influential for me. It was in Scientific American. There was a special issue about the brain, and had all these articles from famous neuroscientists about all we've learned about the brain. And Francis Crick came along at the end. He says, you know what, that's great, we have all this data. He says, but we have no idea what the hell's going on. And let's not pretend we do. And he says, it wasn't that we were lacking a theory. His exact words were, we were lacking a broad framework in which to apply these ideas. You needed a framework for understanding what the brain does in which to apply theories and hang them onto. Now, that was almost 40 years ago, and a lot has happened since then. First of all, we've had tremendous advances in experimental techniques. We're all aware of those. Tremendous advances in the collection of data and the things we can do. The question is, how have we done on developing a broad framework of understanding what the brain does? Well, I think there's been some success and some lack of success. One of the things that's been most successful is the work you just heard about. The whole field of place cells and grid cells and the medial entorhinal cortex and hippocampus. That is sort of a framework for how we map out environments and know where we are. And you can see the excitement and interest that goes into that field right now. But when it comes to the neocortex itself, I argue that there hasn't been much progress in developing this framework. And we still need that, and that's what I'm going to talk about today. Now, there's an artist's rendition of a brain. We all know that's a human brain. Just a reminder that neocortex is this thing that wraps around the old brain. It's about 75% of the human brain in terms of volume. And if you could take that neocortex out of your head, I always carry this napkin with me. This is my model of the neocortex. It's about this big in area if you could flatten it out, which you can't. And it's about 2.5 to 3 millimeters thick. It is basically everything we think about the world, our perception of vision and hearing and touch, mathematics, language, physics, and so on, is all occurring in this sheet of cells. It's quite remarkable. Now, there's two things about the neocortex that are in contrast with each other. One is the diversity of functions, the number of things it's capable of doing, and the other is the commonality of the circuitry. And this has puzzled people for a long time. In terms of the diversity of function, the neocortex is divided into about 100 different regions. No one really knows how many. There are multiple regions for vision and touch and audition and languages, not just spoken languages and written languages, but mathematics and physics, music and art. All cognition occurs there. Those regions are connected in this very complex hierarchy that doesn't really look like a hierarchy, sort of like a hierarchy. And there's this remarkably diverse functionality that has led to our success as a species. But then people have noticed for over 100 years, when you look at the details of the neocortex, there's a lot of commonality. First of all, there's these layers of cells. And then there are these predominantly, looks like the connections that cross the layers are predominant, that information flows up and down across the layers. There's some horizontal connections in some layers. And this is suggested a long time ago that there might be a columnar organization to the cortex. That information flows up and down in the columns and then spreads horizontally. They don't look like this, but that's a theoretical idea. And this is a remarkable similar circuitry everywhere throughout the neocortex and in different species. It is not the same everywhere. There's a lot of differences, but it's also remarkably similar at the same time, and we have to recognize that fact. And it was in 1978 that Vernon Mountcastle was the first one to propose this big idea. And if you haven't read it, you should go read his essay. He's proposed the following. He said, look, I propose that all regions are doing the same thing. And what makes a visual region vision is because it's hooked to an eye, and what makes an auditory region auditory is because it's hooked to an ear. Incredible idea. I put that up there with Darwin's idea of evolution. He said that the diversity of species is all based on one algorithm. And Mountcastle is saying the diversity of our cognitive functions is based on one basic algorithm. He then went on further to say that the functional unit of the neocortex is the cortical column. And that's a debatable thing. What it is? Well, this, for the purpose of my talk today, I'm just going to say take a square millimeter of cortex, and in a human there's about 150,000 of those. And then he said the obvious. He said, look, understanding what the column does is the key problem in neuroscience. If you understand that, then you understand the neocortex, and then you understand 75% of the brain. Sounds simple. But that was his proposal. And it's so radical that today many people still don't believe it. It's like hard to imagine what that could be. But we take that as gospel, acknowledging the fact there are differences, but there's still something common going on here. Now, one of the problems in solving this problem is that the cortical columns are incredibly complex. The circuitry in the cortex is one of the most complex things you've ever seen. And within 1 square millimeter of cortex, there's 100,000 neurons. There's, good guess, about 500 million synapses. 500 million. There's more than 6 layers. There's at least 10. Yes, question?
I'm still puzzled by the previous slide. With this third point, aren't you afraid that understanding the column is going to be sort of like understanding a leaf on a tree?
My concern about understanding a column is understanding a leaf on a tree? No, not at all. I don't see that analogy. It would be more like understanding cells.
And then building from a cell a tree is a very...
So where is the tree in this scenario here?
The whole brain.
The whole brain. Well, okay, the rest of the brain. So are you arguing, am I afraid can we understand the neocortex about the rest of the brain?
I'm saying that understanding a cell isn't first of all necessary and sufficient for understanding that tree. I don't think it's either necessary nor sufficient.
Do you agree that understanding what the cortex does and the column does is a big idea, a good idea?
I think that understanding what the column does maybe is a good idea. I just don't think that it's either necessary or sufficient for understanding what the cortex does.
Well, all the circuitry that the cortex has is contained in here. There's some exceptions. I'm not talking about the thalamus, and there's obviously connections to subcortical areas. But there are no other one of the reasons that Mountcastle picked the cortical column is because every nerve type, every cell type exists in that block of cell types. Every type of connection exists, and basically every type of physiological response that repeats over the cortex is contained in that. So there isn't another place for the cortex to do things. So if you believe in some magic, it's going to happen someplace else, perhaps. But at least you accept the fact that this is a really important problem. And if we can understand that, we should go forward with that. All right, great. Anyway, back to the cortical column. It's extremely complex, and there's at least a dozen or 10 or more cellular layers. There are dozens and dozens of different types of cellular pathways that occur in here that have been documented over the decades. Within the column and in between columns, there's an entire inhibitory cell network, which is even actually more interesting than the excitatory cell network. We acknowledge that there is a significant region-to-region variability in this. There's more cells, we heard that in talk yesterday, more cells in certain levels, fewer in others, more connectivity, and so on. But still, there's something commonality here, and it's been tugging at people for a long time. How do we understand that? Now, you may not like my answer today, but I'm going to walk through what I think is going on here. Okay, now, how do we attack this problem? Our approach is the following. This is what we've done in our research. We start with an observation that the cortex is constantly predicting its inputs. Now, what do I mean by that? When I reach my hand over here, I have an expectation what I'm going to feel. And if I move it up here, I have a different expectation. And when I touch this thing, I have a different expectation. In fact, every time I touch anything, I have an expectation what I'm going to feel. And I know that because if it was different, if this felt like Jell-O or milk or something else, or had a real rough edge on it, I would notice it. So there's this sort of background prediction which is going on. It's happening in vision as well and in audition. And you notice here when your predictions are inaccurate. It's not that your predictions are conscious. You're not aware of this. When we started with this, I wrote an entire book about this problem called On Intelligence, that brain's building a predictive model of the world. And then the question is, how does the cortex, and now, therefore, how does the column learn predictive models of its input? And I argue it does. If it's going to make predictions anywhere in the cortex, it's going to be making predictions in every cortical column, and how it is to do that. So let me just walk through some basic circuitry here, because I want to cherry pick some parts of the circuitry in the cortical column, which I'm now going to try to put a theoretical framework around. So let's just talk about feed-forward pathways into a cortical column. It doesn't matter where you're talking. There are two sources of input. One is directly from other cortical regions, and the other is through the thalamus. This is true, the thalamic input is true not just for the primary sensory regions, but as Murray Sherman and Ray Gillier have argued for many years, it is a part of every cortical column that input comes into every cortical column from the thalamus and directly from cortex if that's available. Now, remind ourselves that when it arrives primarily in layer 4, not exclusively in layer 4, but arrives in layer 4, your input layer in the cortex, it forms less than 10% of the synapses on layer 4 cells. In the V1, it's estimated about 7%. So where are all those other synapses on layer 4 coming from? There's many coming from other layer 4 cells, but about 50% of them are coming from layer 6A. This is a common repeated observation in cortex. About half the input coming to layer 4 is coming from 6A, and I've noted it here in blue because it doesn't appear that those inputs are strong enough to make the layer 4 cells fire. But they must be doing something important. That's a lot of neural pill for just not doing something. So something is happening here in this thing. Then the classic view is that layer 4 projects to layer 3. Layer 3 projects to layer 5. You find this in every textbook. But here you see a similar thing. Between lower layer 6, layer 6B, there's a similar sort of bidirectional connection to layer 5. Those cells look very similar. There's a very unique pattern of how they look. And so there seems to be this sort of parallel construction going on here between 6A and 4 and 6B and 5. This is a very common circuitry. You see this everywhere. There are two types of outputs to a cortical column. There's from the upper layers, predominantly layer 3, but also layer 2. It projects to other areas of the cortex. And then there's another output from layer 5, the thick tufted cells, which projects to the thalamus, becomes then input to another region. And it also, as Murray has argued for many years, it has a motor component. And we've seen this everywhere. So every column seems to have a motor output as far as we know. I want you to remember this part because I'm going to try to develop a framework for understanding what's going on here in the rest of this talk. Now, I'm going to do it using this as my outline for my talk. This represents the outline for my talk, and it also represents the outline for how we've gone about our work over the last 13 years. And we've been trying to tackle this a piece at a time and building up more complex models as we go. We started by asking a simple question. How does a layer of cells learn predictive models of sequences, like a melody? I can learn a melody. I can predict it. I'm always constantly predicting the next note in the melody. Sequences are used everywhere in the brain. We use them in our motor behaviors. We use them in visual inference and auditory inference. It's a common thing that brains have to do. They have to learn sequences and play them back. That took us many years to figure this out. It's a very complex story. We first published it about 2 years ago in March of 2016 in Frontiers. We now have about 5 papers on this particular thing. There's a lot of detail in that, how that works. We then went on to, I'm going to go through some of this in a moment here. We then, after we did that, we wanted to extend that model to how it is that we can learn predictive models of sensory motor sequences. Because inputs to the brain, into the cortex, and into any column, are constantly changing. But most of the time, they're changing because we move. not because the world moves. So we'll call that sensory motor sequences. And we achieve that by doing two things. We deduce that there must be an efference copy or a motor-derived location signal, and I'll talk about that in a second. And we added a second column, second layer in this model. We published that this last year in October of 2017. When we did that and we published this, in that paper, we talked about the nature of this location signal. But we said, look, we deduced that it must exist, but we didn't know where it came from. We didn't understand the nature of it. We speculated in that paper that we could study the entorhinal cortex, which has another mechanism for representing locations, the grid cells, and perhaps we'd find a motivation there for how it is be happening in the cortex. By the time that paper came out, we had already figured this out. We think we figured it out. At least we have a good model for it. We believe that layer 6A is actually doing something very similar to what grid cells are doing in the entorhinal cortex. I call it grid cell-like. It's not exactly the same, but it's using some of the same tricks that we believe are going on in the entorhinal cortex were preserved and actually happening in every cortical column. And now we have a complete sensory motor inference model here. We have now extended it further. We realize that objects in the world are composed of other objects, and I'm going to get to this in more detail. And so we believe that each cortical column is actually learning composite objects. And that you can now explain 6A and 6B as actually two different location layers representing two different structures, and layer 5 is a composite output layer. I will get to that. This has not been published, so we did present a poster at COSIGN this year, and manuscripts are underway. But I can talk about it because we're supposed to be speculative. What I want to get to is I want to talk about what's going on here. But you can't understand what's going on here until I tell you something about what's going on here. So we kind of build this up, and I have to bring you with me. Otherwise, I can't just explain this. So I'm going to go start over here, and we'll talk about how some of the key points and how we think that a layer of cells, and we think even an input layer, layer 4, is learning sequences. We started with the neuron. Where are predictions in the brain occur? Have you ever thought about that? If the brain is making predictions all over the place, how does that happen? You could have a separate population of cells that makes predictions, which is true, but we actually think that every pyramidal neuron is a prediction machine. Now, if you think about a pyramidal neuron, it has anywhere between 5 and 30,000 synapses. 10% of them are close to the cell body, proximal. They can make the cell fire. 90% of them are too far away. And for many, many years, people had no idea what the 90% of the synapses in that cell were doing. Several clever labs have come up, and some of the people here in the room have worked on this. They've discovered that dendrites are active processing elements. And if you have somewhere between 8 or 20 synapses on a dendritic branch, and they're near each other, within 40 microns of each other, and they activate around the same time, then you generate a dendritic spike, and the dendritic spike will travel to the soma and have a large effect. If it was an NMDA spike, it will depolarize the soma, but not enough to make the cell fire. And it can lead to a sustained depolarization. So now, people argued, is that useful because it doesn't make the cell fire? What good is it? We have proposed the following, that the proximal synapses define the classic receptor field of the neuron, but the distal synapses, when they cause these dendritic spikes, they put the cell into a depolarized, what we call a predictive state. The cell is saying, I've recognized the pattern that is typically preceding myself becoming active, and I'm depolarized. And the advantage of that, and this is our hypothesis, that depolarized neurons fire sooner than non-depolarized neurons, and they inhibit other neurons nearby, and they lead to a sparser activation. And I'll walk through how that works in a moment. And a neuron can predict its activity in hundreds of different contexts because they have thousands of synapses. The next thing we have to do to understand this model is, here's a picture of how we model neurons. In our software, everything we do, this is a sort of a picture of how we do it in software. Our neurons have arrays of dendritic branches, if you will, that act like coincidence detectors. As far as I know, we're the only people in the world who build sophisticated networks using model neurons with active dendrites. I don't see how people can feel like they're modeling the brain by using point neurons. I'm going to talk now about sparse activations. This is key to the whole theory as well. If you look at a population of cells, let's say 5,000 cells in one layer in one column, at any point in time, there would be some activation on those cells, and it's sparse. So I'm going to just use an example here. Let's say we had 5,000 neurons, and 2% were active at any point in time, and another point in time, 2% were active, another point in time, 2% were active, just for grins. So we have 100 active cells. A couple observations we can make. First of all, how many different things can we represent with that? It's virtually unlimited. It's basically 5,000 choose 100, which is 3 times 10 to the 211. So almost infinite from our point of view. The next point is, if you randomly chose sparse activations, and you could do that all day, how much would they overlap? How many cells would they have in common? And it's about 2. But what's very important, it's never going to be a lot. It's very, very unlikely that if you pick some random group of 100 cells, that there'd be a lot of cells in common. So you can do this all day long. You can go do it forever. And you can pick these representations, and they're unique. Now, neurons are going to take advantage of that in the following ways. First of all, if I wanted to recognize a pattern in a set of active cells, I only need to form synapses to a subset of the active cells. If I form a subset to maybe 8 to 20 of them, this neuron will fire when that pattern's there. You can argue, yes, but there's a large chance that this thing could have a false positive recognition. It turns out it's not likely at all. It's very, very unlikely. You can do the math on it, and we've done that, and you can also test it empirically. This cell will reliably recognize that pattern in almost all conditions. The second thing, now this is the piece I want to get to on this slide. We think this is going on throughout the brain. In any region, we think that actually what you're representing at multiple times is a union of patterns. That is, you can impose multiple patterns at the same time in the same set of cells. So if I want to invoke 10 patterns that have a little bit less than 1,000 cells active, maybe a little bit less than 20% of the cells active. And what we can show mathematically and empirically, that this cell with its 15 synapses or whatever will still reliably recognize that one pattern it was designed to recognize. There's obviously a much greater increased risk of false positives, but mathematically and empirically we show that it's still very, very small. So what we hypothesize is the following. All of our models work this way, that the way the cortex represents uncertainty is through unions of patterns. And when it's uncertainty, you see higher activity levels. What's going on there, it's a union of patterns. And when activity gets sparser, when the cortex is figuring out the answer to something. And what's amazing is that all the networks I'm about to tell you about, they work with these unions. That is, you can process unions at the same time. You can propagate multiple hypotheses and act on them all through every connection that's going on in the brain. Okay, we put this together in a single layer model to learn sequences and predictions. So you can imagine this like layer 4. Here I show that layer in time 0 and time 1. I've arranged the cells in little mini-columns where the cells in the mini-columns have the same feed-forward receptive field property. It's not important to have mini-columns. It's important to have a set of cells that have the same feed-forward receptive field property. And if an input comes along later, it's going to activate a set of mini-columns, and you'll see all the cells in those mini-columns become active. That's what Hubel and Wiesel saw 40, 50 years ago, and we still see today. What's going to happen now if you have a predicted input? And now I'm going to represent that by these little red circles. These cells are in the depolarized state. The same input comes along. Those cells fire sooner, and they inhibit the cloud around them, and you end up with a very sparse pattern. That pattern represents the input in a specific context. And you can have millions of these. And then that activation pattern can then predict the next activation pattern, and you can basically build up a sequence memory. We have characterized this, so just point out it's very high capacity. Even a small network like this can learn hundreds of thousands of transitions. It can learn any kind of high order sequences. It's extremely robust to everything. The learning is, importantly, unsupervised, continuous, and local. So there's no like global signal going on here. And it really is a biological network. We actually require 2 different types of inhibitory cells to make this work. It's very detailed in that regard. Just to be comfortable, many people have implemented this. There's many multiple source implementations, and it's actually being applied in commercial products today. Okay, that was what went on here. So then we said, okay, how can I extend that model to learn predictions of sensory motor sequences, like when I move? That's a much harder problem. So let me just, I think we all understand the problem here. It's like, this is a static object. And when I touch it, the changes to my brain occur because I move, and I want to be able to predict that. So we started with our same input layer, our sequence memory. We said, how do we modify this to incorporate sensory motor predictions? And we said, okay, the simple answer to that is we need to add a second context. We have one context, which is the cells themselves. We need a second context, which would be some kind of motor-related context. And if we could do that, then maybe we could get the system to do what we wanted to. But what is that correct motor-related context? We struggled this for quite a while. And the answer now was very obvious to me, but it wasn't obvious at the time. And I'm just going to illustrate it with just my coffee cup here. seems to only want to work occasionally. There we go. Literally, I was doing this in my office one day. I said, okay, so imagine I'm just touching this cup with my finger, and I'm about to move my finger and touch it in a new location. And I can predict what I'm going to feel. And I can predict it right now. I know what I'm going to feel. And I know if I touch the bottom of the cup, I'll feel something different. There's a little rough edge down here. And if I know if I move my hand over here, I'm going to feel this little handle here. What does it take for my cortex to do that? First of all, it needs to know that it's touching a coffee cup, because that's essential. And it also needs to know where it is on the cup. And it needs to know where it will be after I move. It needs to know in the reference frame of the cup. It doesn't matter where the cup is relative to my body. It doesn't matter where my finger is relative to my body. It's where my finger is relative to the cup. There's no way around this. If I'm going to make a prediction, I'm going to have to know where it is I'm going to be on the cup after I move. And you can just do this yourself. You can imagine what you're going to feel when you do this. Now, imagine now I'm touching the cup with multiple fingers at the same time in my hand. All parts of my hand, all my fingers are making the same different predictions, but they're doing it. I'm not conscious of it, but they're doing it, because if anything changed, I would notice it. If there was a little rough edge here or something like that, that's not normally there. So this idea that every part of your sensory, your somatosensory area is actually predicting what it's going to feel requires that every part knows where it is relative to the cup. I'm not going to work through it today, but the same argument can be made for vision and the retina. So we came back and said, okay, we need an object-centric location signal to do this. And now I could say, okay, the input layer, what does it represent now? It represents a feature at a location on the cup. And every time I move, that changes. And if I pull those together in a separate layer of cells called we'll just call it the object layer. And I said, this is going to be a stable representation representing the cup. And I'm going to associate that stable representation with the set of features at location. I now have a definition for a cup. And I can predict what I'm going to feel if I have that object-centric location being updated. We have built these models. We've built all kinds of sophisticated versions of them. I'm not going to go through all of that. But this will learn a predictive model of objects through movement, static objects. And therefore, an object is just a set of features at locations. Yes?
So the cups you just used is the 3D objects. So
yeah.
You need to predict the 3D location.
Yes.
2D maps. So do you have
I didn't say anything about 2D maps yet. You said 2D maps.
Oh, okay.
I'm going to argue that the way locations are represented in the cortex are non-dimensional, and they can be applied to 1D, 2D, 3D, ND in a moment. So I didn't say anything about 2D representation. You're thinking grid cells are people say, and that's even questionable whether grid, I think you're thinking grid cells being 2D, but I didn't say 2D. Okay, here's a little cartoon animation to just give you a flavor of what we're talking about here. And I want to show this because I want to talk about the union property a little bit. So imagine I have this finger, and I'm about to touch this cup at some location, and this is my input layer, and this is my output layer, and I have a location signal coming in telling me where I'm going to touch. I get some sensory feature when I touch that, and what's going to happen up here, you're going to form a union of all the possible things that this column knows are consistent with that location and feature in some sense. It says, look, there's three things that are consistent with that: a cup, a can, and a ball. And the way you're going to do inference is you're going to keep moving your finger, just like if I reach my hand to a black box and I touch something, I don't know what it is until I move it several times. What's going on when you're doing that is you move to a new location, you get a new sensory input, and now that is inconsistent with one of these things, and you start narrowing down the representation up here. You do another position, another thing, you get another location, another feature, bingo, now you know what it is. It's the coffee cup. We realize that, of course, multiple columns are being getting input at the same time. So imagine I'm touching this with three fingers at the same time. Each one is going to be on a different location on the object, and each one is going to have a different sensory feature. But they're all trying to model the same thing. And so we realized if you had long-range connections in this output layer, you could form an association memory between these, and they could resolve ambiguity very, very quickly. So an example of that in this guy, imagine now my little thing here. I have the hand touch the cup and three different fingers at once, so these are the three different fingers. Each one's at a different location, different feature. Each one on its own is uncertain of what's going on. Each one can't tell what it is. This guy says, well, it could be a cup or a ball. This one says a cup or a can. But when they vote across columns, they quickly settle on the right answer. This is equivalent in vision. Imagine if you're looking at the world through a straw, and you're trying to recognize something. You have to move it around and to see the different parts. You couldn't recognize it just in one glance, but if I have multiple patches of my retina, the entire retina in some sense, then all of them can vote once and say, oh, I know what this is. We think this is actually what's going on in layer 4 and layer 3 in the cortex. Okay, so that again was published last fall, and I really want to now move over to this guy here and talk about layer 6A, and what I'm going to argue here is that layer 6A is something like grid cells. Now, it was great that Surya gave that very detailed talk earlier, because I'm going to talk about grid cells in a very broad theoretical framework and not go into the details. I'm going to point out a few things about them, and I just want you to trust me that I know the details. I'm just not going to talk about them today. So how it is that, let's talk about how location is represented in grid cells. Now, just remind yourself, grid cells exist in multiple modules. And when you look at the entire set of modules, it encodes a location. Individual grid cells don't. So here's a classic example. You have a rat in a room. Maybe it's a virtual environment, but let's say it's a rat in the room. And what grid cells will do is they will encode different locations in this room. And so what's important here is that these location representations are just, if I just looked at them as a bunch of neurons, they're just some sparse code. And they're unique for each room. This is an important part of grid cell theory, at least in learned environments, that would be the case. And so these two rooms here are similar, but they're different, and the rat will see them different because they have some sort of sensory cues in them which tell you they're different. And so the same locations in this room will be represented by very different activations in this room, and same with this room here. So this is an important thing. These are just the representation of location in grid cells. If I just looked at the population of cells and what's active, represents these locations. They're unique to the room and to the location of the room. So I'm not talking about individual grid cells now again. I'm talking about the ensemble of grid cells. The key thing here is that what ties these points together is movement. If you look at the actual representation of the grid cell population, it tells you nothing about dimensionality. It doesn't say X and Y, nothing like that. It's just some random vector, it looks like. But what's important about it, if the rat moves from A to C, it'll say, OK, I have a new representation for C. And if the rat moves from A to B to C, it also gets back to C. And the clever trick that grid cells do is that once you've learned this, and Surya talked about this a bunch, once you've learned this, it applies to novel environments. You don't have to learn it again. So I can be in a new environment, and all of a sudden I can navigate between these, and the path integration is going to work. One way to look at this is say, what's the definition of a room? A definition of a room is a set of locations, all of them, not just the ones I'm showing here, that are connected together by movement, by path integration. You can move between those points, and the system knows how to do that. Some of these locations have associated features, and that's how you know what room you're in. But not all of them. You don't have to have features at everything here. You just have to have some. What we're going to say is the following thing is going on in every cortical column. But here, what we want to know is, where is the sensor patch relative to objects in the world? And so I can define a set of points around this coffee cup, a set of points around this pen. In the same way as before, there's going to be sort of a grid-like cell representation. We're going to argue it's in 6A. That's unique to the location and the object. So A is only on the coffee cup at this point. And this location updated by movement. And we can say, what is an object? An object is just like over here. An object is a set of locations that are connected by movement via path integration. In this case, I'm just moving a part of my body, not the entire body. Some locations have associated features. Notice I pointed a location W here. That is in the space of the pen. It's not on the pen, but it's in the space of the pen, because if I was down here at Y and I move my finger up here, I will sweep through W to get there. It doesn't mean that you have to be touching the thing. You're just within this point that you can move between. And the way this works, each object has its own point in space, its own set of locations in space. And what you're trying to do is narrow down which location you are. And if you narrow down which location you are exactly, then you know what cup you're on, what object you're on. This analogy can be taken further. This is explaining sort of like primary and secondary sensory cortices. What about higher level concepts in the brain? Could this be happening there too? Well, if you study grid cells, you start realizing that the location representations are dimensionless. Back to your question earlier. If I just look at the representation, it's dimensionless. What defines the dimensionality of the space is how the path integration learns. That's what defines the dimensionality. And so you could have one-dimensional spaces, two-dimensional spaces. It's all about path integration and movement. The second thing is the movements themselves do not have to be physical. If I'm a cortical column and I'm getting some sort of efference copy. It doesn't have to be a physical movement. It could be a movement coming from some other part of the cortex, which would be sort of movement in a different space. And the features themselves do not have to be sensory features. They can be the outputs of other columns. And so what we are proposing is that all knowledge in the brain is represented, even abstract concepts are represented in this way. They're represented in spaces. So when you think about mathematics, the equivalent of a movement might be a transform you do on an equation. It's a procedure you operate on, and then it moves the components to a new point in the space of the problem you're working on. This is a very vague idea here, but I think it follows from Mountcastle's proposal, from the cortical anatomy, and from this basic idea that this idea, if this is happening in sensory areas, it's going to be happening in other areas of the brain as well.
Quick question.
Yeah.
So anytime you're talking about movement, you need to, at some level, start talking in terms of time scale.
Yes.
And is there anything in this model which either predicts or suggests some ruler for the time scale of?
Yeah. I didn't present it here. I'd rather talk about it too because it's a little bit complicated. But you remember I talked about the sequence memory earlier? That has to have time in it. And so does sensory motor inference has to have time. Everything has to be timed. Everything has some sort of temporal pattern by which it flows. Think of it that way. Think of it like the tempo or the rhythm or the individual duration of notes in a melody. And we have a theory about how that's done. And I'm happy to talk to you about it. It's very theoretical, but I believe the current best guess about it is matrix cells in the thalamus, which project to layer 1, have all the right attributes for this, and they look like they might be doing that problem. But it's an important problem, but I didn't talk about it here. Okay. Yeah.
So for grid cells, there is no hierarchy in space, right?
Yeah.
But for objects, there is a hierarchy.
Perfect question. Just lead into my next point here, okay? Object hierarchy. Can I answer that question before I get another question?
I just want to put in a question there regarding the holistic perception. For example, faces, like Mooney faces.
Mooney faces, yeah.
Look through an aperture, you can't really see, eyes, mouth.
Yeah, yeah.
Looking.
Yeah, I think when I talk about hierarchy, let me come back to hierarchy. Okay, if I look through a bigger straw, I could see a Mooney face, right? If I look through a really tiny straw, I might not be able to see the Mooney face. Let me come back in a second when I talk about hierarchy. All right, I think you're asking about compositionality or hierarchical structure.
Because it doesn't exist in space, right? For grid cells, I don't think about any hierarchy.
Let me talk about that. We came to realize that objects are always composed of other objects. This is going to get to the hierarchy here. So here's a classic example. Here is my coffee cup. On the coffee cup, there is a Numenta logo. I've learned this Numenta logo someplace else. And now I know that it's part of my coffee cup. If this wasn't there, I'd say something's wrong. If the logo looked different, something would be wrong. I don't learn the logo again because it's on the coffee cup. I have some concept of the logo. And I have to somehow assign it to the concept of the cup. It turns out that everything in the world is structured this way. Every object is composed of sub-objects. I lied to you when I said that it was features on objects. We don't believe that's true. We think everything in the world is structured by structure of structure, object on object. And if that's going to happen in the cortex, and it does, it has to happen in the cortical columns. And so what we think is going on here is exactly that. We see there's these two, remember there's a 6A and 6B. There's these two parallel structures going on. We think 6B is actually representing, it's another sort of grid cell-like layer, but it's representing the space of the compositional object. And 6A is representing the space of an attended component of that object. So in this case, if I was looking at the Numenta logo, I would be representing it in the 6A and 4, but I would be positioning in the compositional structure of the bigger, larger object. And one of our employees, Marcus Lewis, is here today, came up with a very clever way of how to represent that position of the logo on the coffee cup using another grid cell-like trick. And we think that's what's going on in layer 5. We think layer 5 is actually building compositional structure, and where layer 3 and 4 and 3 are tending to substructure. So again, we've presented that mechanism in that poster. I just wanted to leave you with that because this is a key thing that has to occur in the brain. And now we can start beginning, at least have a hypothesis, a framework for understanding what's going on here. And this has to happen. And once you got it once, that's good enough, because you can keep drilling down. You can look at you can drill on the logo. My logo is composed of lines and words and things. And so you have to do it once, but then you can do the whole compositional structure.
So my follow-up question to
Yeah.
this is, don't you think that your union might break down in this way?
Yeah, it seems like it's like you might get too many. Is that your question?
I'm sorry?
Like you might get too many unions?
Exactly.
Yeah, that's a very interesting question, astute question, which I'd rather take a bit offline. But why doesn't this break down? Because why don't you form unions to form? The answer is a bit complicated. But first of all, the neural tissue doesn't let you get too dense. It breaks. It says, I won't let you represent too many things. There's a certain level of density of activation that you're going to get. And then, so you're never going to get all the cells active at once. And then the system will work. It may take more steps, more movements, but it slowly builds down. It's incredible how even in a very complex system that it settles. And we don't understand all of it, but I will, if you want, I'll talk to you about it later. Okay. I want to take one more slide about hierarchy, then I'll wrap up. This idea of thinking about this brain challenges the way we think about hierarchy. So this is the classic view, if you will. You have some sensory organ, it projects to the primary sensory region, you extract some features, you project to the next region, more complex features, and then eventually you recognize objects. In a deep learning network, this might be 100 levels high. We're challenging that. It's not to say that this isn't true, but you have to think about it a little bit differently. Now we're saying the region actually is a whole set of object models. And the next region up is another whole set of object models. The next region up is another whole set of object models. So how do you think about that? Well, first of all, one of the things you should know that any input to any region always skips a couple of levels too. So there's input from LGN, from the vision, to V1, to V2, and so on. It gets less as you go up. But what that allows us to do is allows the columns at different levels to also work at different scales. So if I'm turning to something very, very small in my visual space, I'm going to argue that V1 is going to be required to do that. But if that thing gets larger, like a letter, and it gets larger, then V1 and V2 could do it. And then if it gets larger still, V1 is no longer able to see that because it takes up it's outside of my, V1's looking at this tiny little space, and this thing is too big, like the Mooney face. So that's one thing to think about. They're working at different scales. The other thing to think about here is that when you have multiple sensory arrays, so now I have like a vision array and a touch array, and so I'm looking at this coffee cup and touching it at the same time. What's going to happen here? You're going to have invoking models in the the somatosensory hierarchy and in the visual hierarchy at the same time. And these guys can collaborate across modalities because they're all modeling the same object. The same argument I made before about long-range connections between certain layers, allowing the columns to vote, applies here too. And what you see in the cortex, you see connections like this that don't make any sense in the classic hierarchy. You'll see connections in a secondary region projecting to a secondary region in another modality, to a primary region in another modality, going up all over the place. That doesn't make any sense in this model, but it makes a lot of sense in this model because these guys can all vote. The corpus callosum can be my left hand and my right hand because those things are often touching the same object and so on. Okay, I'm going to end it with there. There's a bunch of biological. You think, could this possibly be true?
One more question.
Yeah.
For your previous slides, so what
okay.
is the primitive objects on the first layer? Like, and what kind of functions can brain do in order to recognize this very primitive?
What do you mean a primitive object?
So for the objects on the first layer, right after the sensor array. So I assume.
What I'm arguing is that even columns in V1, and I'll talk about it in the next slide, I'll get to this. Even columns in V1 are recognizing complex objects. You don't think of it that way, but here, this thing called border ownership cells came out of Onderhout's lab. They found cells in V1 and V2 that have classic receptive fields, but they only respond in a learned environment. They respond when that part of the visual field is on a specific point of that object. It's as if that cell knows, oh, I'm on the tail of the tiger. I'm supposed to respond. And it doesn't respond in other places you would expect it to respond, but it seems to be tied to various locations. That's a direct consequence. That's showing that even in V1 and V2, there's some sort of knowledge about what the entire object is, and the cells are being unique in location. That's exactly what we would predict. There's another really interesting set of things that are going on right now. There's some very clever experiments going on with fMRI, where they've shown signatures of grid cells throughout the cortex. Not throughout all of cortex, but lots of it. And there's some clever experiments, I won't go into it. It's not proof, but it looks very, very convincing that grid cell-like properties exist through many different regions of the cortex. There's other things, this stuff has been known for a long time, this sort of sensory motor prediction. Even in low-level sensory areas like V1 and V2, there are cells that become active during a saccade before the saccade is finished. They become active before the saccade is finished, and they would become active when the saccade is finished. So after the saccade and there's a fixation, we'd expect that cell to be active. And the cell becomes active during the saccade. It's as if, it's hard to explain that, but if you think about it this way, you say, well, that cell is being given its new location. It says, given this motor behavior, here's what your new location should be, and it has a model of what it's looking at, and it says, okay, I can predict what I'm going to see. Totally explainable to that. There's another interesting angle to think about it from an evolutionary point of view. The people who study the evolution of the neocortex, one of the predominant theories is that there were two older 3-level structures that folded on top of each other to make a 6-level structure. Those older 3-level structures are roughly equivalent to the hippocampus and the entorhinal cortex. And so the idea that you might have, the lower layers of the cortex would be representing locations, the upper layers representing place cells, if you want to think of it that way, and they form this new structure is very consistent with our point of view here. And lastly, I'm going to give you one more observation of this, and I'll be done. I remember when I first learned about grid cells, I was blown away by them. And I was struck by the fact that I never realized that I had cells in my head, my brain, which are representing my location. And it's very easy to figure this out. You can just walk around in the dark, get up in the middle of the night and try to walk to your bathroom, and you have a sense for where you are. There's no clues, no visual input, maybe there's some slight auditory reverberations, but you don't need that. I can say, okay, I'm going to walk over here, and I know now that I'm in front of the screen, and I walk back over here, I'm going to be closer to the lecture, and so on. So I was just blown away that I didn't realize this. And so it's so obvious now. The same thing has occurred to me when I think about the world. You guys look like you're out there. You don't look like you're on my retina. How come I perceive you as out there? How come I perceive my hand at some distance and position relative to my body? I have a continuous concept where everything is located to everything else. And I know how to navigate between everything and everything else. This is what Crick was looking for when he wrote that essay. Like, how do we perceive the world being out there? It's now clear to me that the cortex is actually a location processor. Every single thing that the cortex does is located in space. And you can think about the brain. And so now I think about the cortex as this location processor, and it uses sensory input to figure out what locations it's in. It doesn't process sensory input per se. It uses sensory input to decide where you are. And it models objects in spaces based on how they move within their own spaces. So that's my end of my argument there. I was speculative. I met my goal. And I want to thank, this is the entire team at Numenta. This is a group of myself and plus these other 5 people, some of them here today, sort of the core research team. This is a collaborative effort. We do this all day long, well, most days, and write these things, build models, and test them. Thank you very much. Yeah, Bill.
I love hearing you, Jeff, because you're like the only person who's a hardcore hardware engineer and a grand theorist at the same time.
Oh, thank you, Bill.
And it seems like we think alike in a lot of ways, because I've been on the 3-space thing for several years. The difference is, when it sounds like you're building your model partly out of the mathematical question of what it takes to represent space, what kind of information flows are necessary, because I see your instincts for we have to have a signal from here, we have to have this, we have to combine them this way. But on the other hand, you're also looking at what is in cortex, what do people measure, what are the cells, what are the animals and stuff. A few years ago, I just did a clean room install and asked about the math only. Like, what ideal brain would exist on the other side of the universe for representing space?
Is that the paper you sent me?
Yep.
Yeah.
And
It's quite different.
Now I'm wondering.
I don't think we should try to explain it to this group right now.
I'm wondering if you can take up what you're doing here. Which parts do you see are essentially mathematical requirements imposed by Mother Nature, and which parts are things we happen to have measured? Which is a theory and which is the experiment in what you're saying? Because you've got a model of
You're saying which of these things are a conjecture and which are...
which things are as nature would do it if she could build anything?
I never think about it that way. I never think about what is mathematically expedient. Physicists think that way. I don't think that way. I look at the anatomy and I say, that's what it is. Okay. And I don't ask, is it the best way? I said, that's the way it is. We want to understand that. There may be much better ways of doing this. I don't know. Our goal, I said this up front, our goal is not to look for biologically inspired theories. Our goal is to explain the neuropill. And I'm going to end it at that. Other people, physicists, perhaps yourself and others, can say, oh, what's the better way of doing this? We don't start with the math. We actually apply mathematics later. I didn't show any of the equations here. We can talk about this stuff. But to me, it's all about starting with the anatomy and physiology and sort of deduction about what must happen, and then try to figure out those things. Yeah.
So the cells in MT, right?
Yeah.
People first solve the aperture problem through intersection of constraints to find the global motion.
Yeah.
Those cells also belong to this stuff?
I don't know that work. I think of MT as part of sort of the aware pathway in the visual system, right? I'm not familiar with the work you're reciting there, so I can't comment about that.
The aperture problem where an object is moving, V1 cell will think of the edge to move exactly in the perpendicular direction to the edge, but the object is actually moving some other direction, and the question is how does that problem solve?
I don't know. I can't answer that question. I can't answer this, for example. I didn't mention this in the talk, but it's somewhat related. It's been noted, of course, there are what and where pathways in vision, the dorsal and the ventral streams, but there are also, they've been seen in audition and somatosensory as well. So the argument has been made that this is a constant theme. And this algorithm, or this column has to solve both. The way I think about that, I was describing here right now was a what pathway. It's like modeling objects in space. And the where pathway equivalent would be in my body space. So I'd be modeling structures around my body space, so I know where things are relative to my body, where my different parts of my body are relative to things, but it wouldn't tell me what they are. And so classic, when if you damage the dorsal pathway, the where pathway, you can recognize objects, but you don't know how to reach them. If you damage the ventral pathway, you can't recognize objects, but you can reach them and be surprised that you reach them. So I think the same algorithm will work in both cases. It's just a different spaces, different sort of spaces they're working in, and we haven't worked that out in any detail. And I can't answer the question you asked me. I'm sorry about that. Yeah.
One more question. So thank you for your talk, and I understand the concept of objects can compose more complex objects. But my very first question is, how do you think the brain can come up with, recognize the very first objects?
How do the brain can recognize the very first object?
Yes.
Well, it doesn't recognize the very first object. It has to learn the very first object.
Right. What kind of, is there any mathematical functions that we can use here? Like for example, in deep learning, basically people use linear functions, activation functions.
Yeah.
Do you think that's enough, or do you think?
We don't think along those lines with deep learning. We think along I didn't talk about the learning rules in our neurons. We rely on synaptogenesis and silent synapses and other things going on. All the learning rules are sort of biologically, very deeply biologically inspired. But we don't sort of satisfy some sort of energy function, or that's not what was going on. There's no back propagating error signals. These are all local learning rules. Everything we do is local learning rules. And so essentially, if you want to ask, how do I learn something new, it's pretty simple. In a very simplistic way, if I just said, I don't know where I am, I could just start assigning myself a brand new location. I could say, just pick a location. This is some new thing. I don't know what it is. Pick a location and then start building a map around that location. I can be simultaneously trying to recognize this as something I know. In fact, we actually believe this is going on. You're actually constantly building new things at the same time you're recognizing old things. The learning and the inference occur in parallel.
So basically, if you look at left and right eyes, so basically we perceive the world as attributes, like for example, red, green, or blue.
Yeah.
So those are just numbers, right? So how do you think our brain can form those numbers into the concept?
First of all, if you took two eyes, you mix up two things, two eyes and then color, right? A cortical column doesn't know what its inputs represent, has no idea. The axons look like axons, sodium spikes look like sodium spikes. That's all there is to it. And somehow you're asking, well, where does color come from? There's been some great work on this, and I'm trying to remember the guy's name. He wrote the book, Why Does a Bell Not Sound Like Red, or something like that? What? Oh, there we go, right here. Sorry. There he is. There's the man. If you want to understand that problem, read his book. Yeah. I'm sorry. I didn't know you were here. Yeah.
Seems your elemental thing, when you were starting out on this, I think you were focused on temporal sequence prediction.
Yeah, this was where we started.
Like most of this is more in the paradigm of constraint satisfaction, perhaps, because I mean, you could look at it as the temporal or sequence prediction is basically kind of a constraint propagation, and you're predicting. If you look at it from a structural standpoint rather than a temporal standpoint.
I don't understand that. How can you look at it without a temporal standpoint? I'm missing that.
Well,
sequences are temporal by their nature, right? That's the whole point of it.
Well, I'm just saying it doesn't necessarily correspond to the temporal information in what you're perceiving.
Well, my speech is a temporal structure, right?
Yes.
And am I being given the hook here, Wolfgang? Yeah. My speech is a temporal structure, and the order. Someone's got like a.
That's my phone.
That's your phone, huh? maybe it's commenting on your question. I don't know. Like, Siri is smarter than you think.
Sorry, I can't find my phone.
Yeah, I'm not sure what your question is. We understand that when I talk about sequence memory, I was talking about ordered sequences. And sensory motor sequences are not ordered; they can occur in any order. But the temporal structure is what's essential there, and we don't view it as we're just trying to learn that temporal structure. What follows what follows what follows what, and how can you do that in a very robust way? And I didn't go through. I have a whole talk so I can do about the sequence memory. It shows how it's really clever, how we can decide, can track multiple sequences at the same time and make multiple predictions at the same time, and sort of like recognize subcomponents of other sequences and so on. But I don't understand your question. So, well, I can talk about it. Yeah. There's a number of us here today from Numenta, so we'll talk about it. I think I'm supposed to. I don't know if I keep going. One more question.
I think we should stop here.
Oh, we stopped. I'm sorry. So, look, I'll be around all today and tomorrow and the next day. So, and a number of us here too, if you want to talk about it. So, thank you again.